What's good, y'all, and welcome back to another edition of Mr. Fitzgerald's biology class. Today we're going a little bit deeper into the cell division world by talking about meiosis. So previously we've already talked about the cell cycle and mitosis. Now we're going to get some background information and start comparing and contrasting mitosis to meiosis, the two different major types of divisions that occur in the cell. So before we start, it is important that we identify the differences between cell division, mitosis before we talk about meiosis. So having you review to understand which ones are associated with which will help you get a better understanding and compartmentalize your information in your brain. So remember, the cell cycle is a process where a cell is growing, it's replicating or duplicating its DNA, making another copy, and then it divides into two identical daughter cells. So this whole process is like the life cycle of the cell for normal growth and division, okay? Now, mitosis is, remember, when the nucleus divides and the DNA is being divided into those two daughter cells. So it's not the whole process of division, it's actually only division of the nucleus itself. So the phases with that is our PMAT, our prophase, our metaphase, our anaphase, and our telophase. Prophase is where we get that DNA to condense down into its chromosomes. Metaphase, where we line them up in the middle. And then anaphase, where we rip them apart. Those chromosomes get pulled apart. And then telophase, we get two new nuclei on opposite ends of the cell. Okay. So what are the two major events that occur during interphase? Hopefully you remember that interphase is composed of G1, S, and G2. The G's, remember, stand for growth. So G1, the cell grows in size, right? It's getting bigger, it's accumulating mass, it's building proteins, it's doing normal cell functions overall. And then G2 is when it actually increases its number of organelles. And that's right before the mitosis begins in cytokinesis, so those division stages. You need to double up your organelles, have another endoplasmic reticulum, another Golgi apparatus, more mitochondria, and all that other stuff. So that way each cell gets what it needs at the end. Also, in the S phase that happens in between G1 and G2, that S phase is where the DNA is synthesized into a new copy. Replicate or copy the DNA, so we have double the DNA. This ensures when we divide the cell, we have equal parts on both sides through mitosis. Okay. Now, what's happening to the parent following cytokinesis? Well, the old cell actually splits into two identical daughter cells. So after mitosis has occurred, where the DNA and the nucleus has been divided, two new nuclei form, the cell body splits, resulting into two smaller identical cells that now start up that cell cycle again and go back into interphase with G1. They grow, they replicate their DNA, they duplicate their chromosomes within the cell as well, or duplicate their organelles, excuse me, and then they start their own mitosis and cytokinesis to keep the process going. So, what happens to a cell if it's dividing uncontrollably? This is a question that usually stumps a lot of students, but you actually probably know the answer to this. If a cell is continuously dividing and nothing is telling it to stop, you'll probably get too many cells in an area, and this is usually called a mass. And when we have a mass that forms, often this is a signal of cancer, especially with a large organism like a person. So cancer is actually some sort of change or mutation in the DNA or the proteins that regulate cell division, where they're not telling it to stop. So a very simplistic view of cancer is it's basically just the cells are dividing and they're replicating constantly and nothing's telling them to stop. They can have different versions. There's benign cancers, which is just a localized tumor usually that can be kind of cut out or removed, and there's no harm to the organism that comes from that. Or there is malignant, where parts of that mass are breaking off and they spread throughout the body, and then it has that uncontrollable growth in different sections of the body simultaneously, which can have very negative consequences for the organism, right? So, we're going to start talking about meiosis and its backgrounds, okay? So now that we've discussed cell division again, we've discussed meiosis again, now we're going to talk a little bit about meiosis, okay, to see the differences. Just to give you a visual comparison, just to wrap up mitosis a little bit before we move on to meiosis. Again, you're duplicating the cell. So you see the cell on the far left there, it is the exact same as the two cells over here. So we have the DNA that's duplicated in the chromosomes here. We divide those chromosomes up, we split them apart, so that way the sister chromatids move to each side, and then we get two identical cells as a result. Meiosis is a little bit of a different process, but to get into it, we got to get some vocab done first. So a lot of times with meiosis, we're actually going to be talking about genes and variation with genes, because this is what leads to sexual reproduction. So the traits that we have that make us us and what we think about is mostly called our genes. Now, our genes are not the exact thing that tells me to have blue eyes or me to be tall. My genes give information to make proteins, and those proteins allow me to be tall or have blue eyes or have brown hair or whatever, okay? So all those different things come from what the information is on the DNA, called the genes, and they're the ones that make specific proteins, and those proteins are what tell you what makes you you. Now, as a human, you have 46 chromosomes that are arranged in 23 pairs. We're going to get on the next slide here, but you also have thousands of genes. We're still kind of working through and sequencing the human genome. We've had actually some success in seeing some genes and proteins that do certain things, and we can identify them early on in a person that they have these proteins and they might actually develop this disease. They might have a higher susceptibility to some disorders. So we actually are using that information productively to find out more about us and how we can actually predict things about our offspring and stuff like that. So it's kind of some cool research that's going on there. We have, again, 46 chromosomes that are arranged in 23 pairs. And how do we get 23 pairs? You get 23 chromosomes from mom and 23 from dad. So mom provides the egg cell, dad provides the sperm cell, and that's what's holding the two pieces of information that make you, or that make the animal, or the plant, because a lot of species use sexual reproduction. It's not only humans. Trees, for instance, produce pollen, that's their sperm, and they have egg cells within either cones or within their flowers to represent that kind of sperm and egg mentality as well. So it's all kind of the same no matter what species you talk about. We just use humans a lot because it's what we are, so we're most familiar with that. Now, these chromosomes are similar, but they're not identical. So when we have the 23 pairs, we call those homologous chromosomes or homologous pairs. The prefix homo actually means same. So when we're talking about homologous, what we're meaning is same-ish or kind of similar. So they're similar chromosomes, but they're not identical. So you don't get identical DNA from both mom and dad. If you look at your mother and your father, they're probably different. Or if you look at any two people, they probably look different, except for identical twins. And we're going to talk about that a little bit later. But usually you have some same DNA, like some similar parts, but some parts are a little bit different. So what that means is it gives us some different gene expression. For instance, let's say that this purple one came from mom and the green one came from dad. These genes here that are on these chromosomes might give you like your eye color or your hair color. Okay, so let's say it's eye color just to stay consistent. Maybe mom gave you the gene for blue eyes and dad gave you the gene for brown eyes. They're both containing genes for eye color, so that's why they're the same. But they're different variations. One's blue and one's brown, so the same-ish, right? It's not perfect, but it gives us some variation. This is how we can have different appearances, different features within our populations, and have us look different. If I have a sibling that I don't look identical to, we're 18 months apart. I have a half-brother that I really don't look identical to because he's mixed with another version of DNA from a different mom. So we have all that variations in our genes, which allow for different proteins to be expressed and allow us to look all different in traits. Okay, so meiosis, if I were to give it a definition, is the separation of homologous chromosomes. So we're going to separate those same-ish chromosomes. We're not going to duplicate and rip chromosomes like we did in mitosis. We're going to separate those same-ish chromosomes so we give a little bit of some DNA to each new daughter cell that's created. When we have those cells that are made as a result of meiosis, like these things called gametes here, and gametes are just another name for a sex cell or a sperm or an egg cell, right? And so these gametes actually have half the DNA in them with some variety, so they can be mixed up with another gamete. Let's say a sperm is mixed with an egg cell here, and that makes a new organism that has a variety of DNA. This DNA coming from this source, this DNA coming from this source. Now we have a combination of DNA and different traits in here as well. We also get another name for these gametes called haploids, and that's easy to remember. If you look at the gamete, it's only got one chromosome in it, for this example. If you look at the new organism, that has two in it, okay? So haploids are called half, right? It's got half of whatever the organism is. So in this case, the organism has 2 chromosomes, so the haploid only has 1, okay? Now, if humans have 46 chromosomes that are in 23 homologous pairs, each pair contains similar but not identical DNA. Why do you think the DNA is not identical? Hopefully you thought back to earlier in the slide where you think of where we get these 23 chromosomes from. You get 23 from mom and 23 from dad. So your DNA is not identical because your parents are not identical. They're not clones of each other, so you're not a clone of them. You're a mix and match, a variety of all the DNA in both of their histories. You might have some features that were in grandparents or great-grandparents. Things might skip generations, and you might actually get those features that somebody else doesn't within your family. So you have this large variety of information that's in you that's related to all the main features like hair color, eye color, height, stature, metabolism, all these different things, but you might show it differently than others do in your family because you have different proteins that are being produced based on the genes that you inherited. So, looking holistically at mitosis and meiosis, just real quick to kind of set the stage a little bit for tomorrow when we talk about the stages or steps within meiosis. Meiosis is going to be a 2-division process, so I'm going to actually break twice. Notice there's a division here, and there's a division here, okay? Also notice we end up with 4 new cells at the end, so these are called the daughter cells. And also notice it has 1 end here instead of the 2 ends, so we call this a haploid cell. It's got half of what we started. 2 is double of 1, so 1 is half of 2, okay? On the other side, mitosis, we're making clones. So notice the end product in the beginning are exactly identical here, okay? So we only have 1 division that results in 2 cells. And that's all I got, honestly. So stay tuned for tomorrow's video where we're going to talk about the steps of meiosis and actually break down the cycle that it goes through as well. If you have any questions, feel free to reach out via Canvas, and have a great day.